You have a morning light routine. You step outside, or you sit by the window, or you switch on that bright lamp within the first few minutes of waking. You have read the research. You know that light in the morning sets your circadian clock. You are doing the right thing. Except there is a good chance you are setting the wrong clock. Not because the science is wrong. The science is solid. Light in the morning is one of the most powerful circadian signals available to you. But the way most high performers apply that signal, the timing, the source, the duration, the sequence, means they are often reinforcing a rhythm that is slightly out of phase with their actual biology. And slightly out of phase, compounded across weeks and months, produces exactly the kind of low-grade cognitive erosion that no amount of optimization can fix. This is what that looks like in practice. You wake up, you feel groggy, you push through, you get your light, you have your coffee, you sit down to work. By mid-morning, you are firing. By early afternoon, something has gone quiet inside you. By evening, you are inexplicably alert at the exact moment you need to wind down. You tell yourself it is stress. It is deadlines. It is the coffee you had too late. It is not. It is your clock. And it has been set incorrectly since morning. Here is the mechanism you need to understand first. Your body does not run on one clock. It runs on a hierarchy of clocks. There is the master clock, the suprachiasmatic nucleus, a tiny paired structure sitting above the optic chiasm in your hypothalamus. It contains roughly 20,000 neurons, and it is the conductor of your entire biological orchestra. But below it, in almost every tissue in your body, your liver, your lungs, your muscles, your skin, there are peripheral clocks, each one tracking time, each one capable of drifting. Satchidananda Panda at the Salk Institute in California has spent years mapping the relationship between these peripheral clocks and the light-dark cycle. What his research makes clear is that the master clock is exquisitely sensitive to light. But the peripheral clocks respond more strongly to other cues: food timing, body temperature, physical movement. When these two clock systems fall out of sync with each other, the result is what researchers call internal circadian misalignment. Your master clock says one time, your liver says another, your muscles say a third, and your brain, trying to coordinate all of them, pays a performance tax for every hour that misalignment persists. So when you focus exclusively on morning light and treat it as the complete answer, you are addressing the master clock, and that matters. But you may be leaving the peripheral clocks entirely uncorrected. Now, this is the point where most circadian content would tell you to simply add food timing and exercise timing to your protocol, and everything will resolve. That is not wrong, but it misses the more immediate problem with the morning light routine itself. Your light routine may be setting even the master clock incorrectly, and the reason is one that almost nobody discusses. The suprachiasmatic nucleus does not respond to light in a linear way. It responds through a process called photic entrainment, mediated primarily by a specific class of photoreceptors in your retina called intrinsically photosensitive retinal ganglion cells, or IPRGCs. These cells are maximally sensitive to short-wavelength blue light, peaking around 480 nanometers. They are slow to respond and slow to saturate. They are not the cells you use to see. They are the cells you use to tell time. What this means practically is that the timing of your light exposure relative to your current circadian phase matters far more than simply getting light early. Charles Czeisler at Harvard Medical School, whose work on light and human circadian rhythms spans 4 decades, has demonstrated that light delivered at the wrong circadian phase can shift your clock in the wrong direction, or fail to shift it at all. Here is where the routine breaks down for most people who are doing everything right on paper. If you are someone who naturally runs a slightly delayed circadian phase, and a substantial portion of the population does, particularly those under 40, your biological dawn is not occurring at the same time as the clock on your wall. Your melatonin suppression, your core body temperature minimum, the precise biological markers that define where you are in your rhythm, may be running 60 to 90 minutes later than your alarm. Which means the light you are delivering in the first 10 minutes after waking is hitting a clock that is not yet ready to receive it as a morning signal. You are not wrong to get morning light. You are wrong about when morning is. This is not a small distinction. Anna Weisser at Washington State University, researching circadian phase and sleep homeostasis, has shown that individuals who consistently receive light signals at odds with their endogenous phase experience measurable impairments in prefrontal function, the exact cognitive territory that ambitious professionals depend on most: attention, working memory, inhibitory control. the things you are trying to optimize. So what does a correctly timed morning light routine actually look like? The first step is not getting more light. It is understanding your actual phase. The simplest proxy available to you is your natural wake time on days with no alarm, no obligation, no social pressure. Not what time you wish you woke up. Not what time you wake up under duress. The time your body chooses when left entirely alone. That time tells you where your phase is anchored. If it is consistently 7:30 or 8 a.m. rather than 5:30 or 6 a.m., your biological morning is not the same as your scheduled morning. Once you know your natural wake time, you can begin to understand whether you are delivering light before your temperature minimum, the point roughly 90 minutes before natural waking when your body begins its ascent toward peak morning alertness, or after it. Light delivered before your temperature minimum advances your clock. Light delivered after it has a smaller but still meaningful advancing effect. What you want, if you are trying to shift your phase earlier, is consistent light exposure in that pre-minimum window. But if your alarm is pulling you up 2 hours before your natural wake time, that window may be occurring while you are still in bed. The practical adjustment is not to abandon your morning light routine. It is to be more precise about what you are trying to achieve with it. If you are running a delayed phase and you want to advance it, to genuinely shift your biology earlier rather than just dragging your body out of bed on borrowed cortisol, the protocol is sequential, not instantaneous. Bright outdoor light, ideally 10,000 lux or higher for 20 to 30 minutes, within the first 30 minutes after your natural wake time. Not your alarm time, your natural time. Repeating this consistently across several weeks, combined with removing light in the 2 hours before your natural sleep onset, will begin to move the anchor of your phase forward. Gradually, without the fragility of a routine built on willpower rather than biology. If your goal is not phase advancement but phase maintenance, you wake naturally at a consistent time, and you are simply confirming that signal daily, then your existing routine is likely working, and the refinement is intensity and duration. Most indoor light sources, including many artificial morning lamps, do not reach the threshold that IPRGCs require for robust entrainment. You need outdoor light or a clinical-grade light therapy lamp at full distance for a meaningful portion of your morning window. There is one more element that most morning light protocols omit entirely, and it may be the one that matters most for the peripheral clock misalignment we discussed at the start: the first meal of the day. Panda's research and subsequent work from his group published in Cell Metabolism has shown that the timing of the first caloric intake is one of the strongest entrainment signals for peripheral clocks, particularly the liver clock, which regulates glucose metabolism, cortisol sensitivity, and the hormonal cascades that underpin your cognitive performance through the morning. If you are getting morning light at 6 a.m. and then eating your first meal at 11 a.m., you are sending your peripheral clocks a contradictory signal. Your master clock is being told it is morning. Your liver is being told it is late afternoon. The mismatch is not abstract. It shows up as inconsistent energy, attentional variability, and the kind of mid-morning cognitive dip that feels inexplicable when you slept well. The protocol that brings these two signals into alignment is straightforward. Morning light followed within 30 to 60 minutes by a moderate first meal. Not a large meal, not a coffee, and nothing else. Something with sufficient protein and fat to activate the peripheral entrainment signal without producing a glycemic response that steepens the post-meal trough. This combination, light then food in sequence, communicates the same time of day to both the master clock and the peripheral system. That coherence is what stability of energy and cognition across a full working day actually feels like. The next time your alarm goes off, do not immediately reach for the light. Give yourself 60 seconds. Ask what time your body thinks it is. Then go and find the brightest light available to you, outside if possible, and stand in it for 20 minutes. Not looking at your phone, not reviewing your calendar. Simply receiving the signal your biology has been waiting for. Then eat something real. If you want to take this further, to build the full pre-sleep and sleep architecture that makes the morning signal land correctly in the first place, the 15-minute sleep shift lays out the complete nightly protocol, 15minutesleepshift.com. The morning begins the night before. That has always been true. Now you have the biology to understand why. You stepped outside this morning to set your clock. The question was never whether light matters. The question was whether the clock you were setting was the one that needed setting, and whether the time you were setting it was the time your biology recognized as morning. Those are the same question. And now you have an answer.